Good morning, everyone. Very good, very good. I would like to welcome you to the 4th Annual Cancer Health Disparities Symposium. We've been holding this conference for the last 4 years, and I would like to thank a number of people. The first is Dr. Lee. Where are you, Dr. Lee? For leading the group. Thank you very much, Dr. Lee. Dr. Lee is from Stony Brook. We are a collaboration between SUNY Stony Brook, SUNY Downstate, and Cold Spring Harbor Labs, and we are sponsored jointly by the National Institute of Health to bring this program together. So we're really very thankful for your leadership in trying to bring the collaboration together and leading the NIH way and making sure that everybody is in line and trying to address the issues of cancer health disparities, particularly GI cancers. I also want to thank our program advisory committee, Drs. Carruthers and Baines. They are coming from Michigan and from North Carolina, and they've been with us for the last 4 years trying to help us build the programs, and we're so thankful to you for being here every year, same time, and providing the guidance that is necessary for us to move forward. So thank you very much. I would also like to thank our institutions, the various presidents of SUNY Downstate, Stony Brook, and Cold Spring Harbor. With us this morning is our president's representative, the Chief of Staff, Dr. Kidron Gwynn, to make some remarks before we start.
Good morning. How are you? Good morning. Good morning. I know it's early. I know it's early. I know how it is. It's snowing outside. Nobody really wants to be here today. I understand. But this is a very important program and one that we are so happy that at Downstate, Morrow and his team have done an amazing job bringing it together. Our partners at Stony Brook, we're just happy to have you guys here. We hope that you guys will stay for the entire day. Get all you can out of it, take it back. I'm glad this is not happening in February, because of the theme of the program. We don't want this to be confused as something that's associated with Black History Month. Granted, it is Black history. We should know that. It is Black history. But the sacrifice, the unauthorized sacrifice that Henrietta Lacks made is certainly one that is beyond Black history. So I'm glad that this is sort of symbolically on March 1st instead of in February. So I'm just glad that this is a program that we can all enjoy, something we'll take a lot from, and again, welcome to Downstate.
Thank you. And now calling our Dean, Dr. Michael Lucchesi, to make some remarks.
So good morning everyone. Dr. Gwynn and I were just sort of arm wrestling and see who was going to follow who, and I didn't want to follow him and he didn't want to follow me, but he's much bigger than me, so I lost. So I want to thank Morrow, and I want to thank everyone who put this program together. It's amazing to me that we're in Brooklyn, the best borough in New York City, the greatest city in the world, right? And here we are talking about health disparities. So shame on us. Shame on us. Shame on us. It's really incredible. This is 2019, and we're still not delivering the kind of care that is delivered elsewhere. And it's just something else. As a healthcare provider and somebody who's been in this business for 30 years, it's embarrassing. So it's extremely important to talk about this, and it's extremely important to spread the word and to really take care of everyone. Everyone is entitled to health care, and everyone should get the same wonderful care. We're the greatest nation in the world. It's just terrible that there's healthcare disparities. And it's not just cancer, it's everything. So welcome, enjoy. It's a great lineup of people and speakers, and I'm very proud to be here, and I'm very thankful that Morrow invited me to welcome you all and give some comments. So have a great day.
Thank you. Thank you very much. As a background, you all know that cancer is very serious business. A lot of people are dying, and particularly African Americans are dying at a very, very high rate, and we don't understand why. And so this group's job is to get to the bottom of these kinds of answers as to why there is these biologic differences, incidence differences, and all those modifications that allow the one group to get more disease than the other group. So that is what we are actually trying to get to. And it's a slow process, but it's a very sure process. We've been working together for 4 years now, funded by the NIH. The team is really very serious. The reason we have this collaboration, because the collaboration, each of us brings a different kind of strength to the collaboration. Downstate and Kings County Hospital, we bring the population to the equation. Stony Brook brings the science and then the population of clinical specimens from white population. So you have a campus that brings mostly, Caucasian population samples, and a campus that brings mostly African American samples. Then we can make the comparisons and see what are the biologic differences that account for these problems. And then we have Cold Spring Harbor Lab. That is our, science home for all these research activities. So I think it's a beautiful collaboration. We want this collaboration to grow, and hopefully we'll be able to provide answers as we move along. Every year we have this conference. I see some of the faces that come to the conference. We thank you for always coming and participating in the conference. We hope that you'll enjoy the rest of the day. Please stick around. I see some fellows here from hematology oncology. That is part of the curriculum. I think we would have a number of the programs, residency programs, fellowship programs actually coming as we move along, so that's also very good. So in the end, this is supposed to promote the academic discussion. In the end, it's supposed to be networking, engage everyone, so that we can all be involved in the conversation in the hope that we can create new ideas, new programs, new research projects. That is what today is all about. So in the interest of time, I'd like to thank you again for coming. And I'll now invite, is Jenny here yet or not yet? Otherwise, I'll start introducing the first speaker. She's not here yet. Okay, very good. All right. So, so then I'll take her role and then play the role until she comes. So we have divided the day into 3 sessions, right? So session number 1 is diet and cancer. So we're going to explore how diet and cancer influence how diet influences cancer. That's what we're going to explore in the morning. And then we would have our keynote speaker around 10 o'clock. And the keynote speaker was actually here yesterday to present himself in an open forum to the campus. A lot of people came. His name is Alfred Lax Carter. He came with his wife, and they did a marvelous job of telling their own story of the Lax story, which is everybody's story now, whether you're black or white. That story is an American story, and it's good for everyone. So he was here yesterday, and it was amazingly emotional and very, very nice. And we all enjoyed it. So he's going to be the keynote speaker for this year. And then the next session is going to be pancreatic cancer, and the last session is going to be on colon cancer. So that's how we've arranged the day. Okay, so on this first session, we would have our first speaker. Ms. Obeid is right here. Okay, good. So let me say something about Ms. Obeid. Dr. Obeid, I'm sorry. You can see that I'm taking Jenny's role now. Okay. So Dr. Obeid is professor of medicine at SUNY Stony Brook. She received her MD in 1983 from the American University in Beirut, Lebanon. She did her internal medicine residency program at Duke and a fellowship in endocrinology and geriatrics, and some work at Duke at the same time. She rose through the ranks at Duke to become associate professor of medicine and cell biology. And in 1998, she was recruited to the Medical University of South Carolina in Charleston, where she served as the Boyle Professor of Medicine in the Division of Geriatrics, and as Professor of Biochemistry and Molecular Biology and Staff Physician at the Charleston Veterans Affairs Hospital in 2012. She was recruited to become Professor of Medicine and Dean of Research at Stony Brook University in 2017. Is that correct? 2012, okay. So she launched her research career by studying the role of signaling lipids in cell stress. So she's going to talk more about how these lipids stress out cells. So that's a very good topic. So we're very happy to have Dr. Obeid, and her topic today is going to be on bioreactive sphingolipids, how they play a key role in cancer metabolism. So thank you, Dr. Obe, for coming.
Good morning, everyone, and thank you so much for the kind introduction. I know Jenny's on her way. They texted me, and it was really bad traffic this morning. So we came the night before and stayed at a bed and breakfast, which was really nice and smart idea to do. Anyway, good morning. This is a beautiful building. It really is, and I'm so happy to be here. And I know I'm happy to see Dr. Ian Taylor, who is the person who recruited me to South Carolina from Duke. We were friends at Duke, and then, so good friend and great person. Okay, so the talk today is going to be about bioactive sphingolipid metabolism, and I changed the title to The Plot Thickens to make it more on cancer, but I sent my slides before I made many changes. So at any rate, basically, the talk is going to be about these lipids that we work on that are very important in cell regulation, and we've uncovered a really interesting role for them in cancer. People don't think of lipids as associated with cancer, but over the last 2 decades or more now, I think this pathway has become really well studied in cancer. So what I'm going to talk about is this bioactive lipid pathway. And it looks a bit scary when you first look at it, but the basic what's amazing about this pathway is it starts by the condensation of a very basic amino acid that's very important in diet and also important in cancer, as I tell you in a bit, and palmityl-CoA, which is a fatty acid that is, again, very important in diet and in many, many related diseases. So these two molecules, palmityl-CoA and serine, start condensing together to form the backbone of sphingolipids, which is this basic sphingoid, 3-ketosphingolipid, which is then quickly reduced to form dihydrosphingisine, and then it's acylated, so another fatty acid is added to make dihydroceramide. And it's an interesting pathway, but the bottom line is you get to this molecule, ceramide, which is a very bioactive molecule, and we implicated it in cell death, in cancer cell death, more than 20 years ago now, and it's really opened, us and others opened a really nice field to study this pathway in cancer. So ceramide in cells has one of two fates, one of several fates. It either becomes phosphorylated to make another bioactive lipid, and the blue ones, all the blue molecules are bioactive, meaning they on their own have biologic activity. So sphingosine phosphate is a pro-inflammatory lipid, whereas ceramide is a pro-cell death lipid. Ceramide can also be made into sphingomyelin in cells, which is 10% of all cell membranes have sphingomyelin, and it's a very important molecule because when it's hydrolyzed back to ceramide, and the enzymes that regulate its hydrolysis are implicated again in cell death. Ceramide can also be modified by glycosylation to make glycolipids, which are again very important molecules that sit in cell membranes and form places where receptors can interact, or many of the complex sphingolipids can be important in immune cell responses. Ceramide can also be broken down by a ceramidase. There are many of these enzymes, and to make a bioactive molecule called sphingosine. And these sphingolipids are named because with the sphingo thing in the front, meaning they were enigmatic lipids, and it's based on the sphinx, people didn't know what these lipids did. So I think we're slowly solving the enigma of the sphinx by understanding these molecules. Sphingosine can then be phosphorylated to make by sphingosine kinase, a very, very important and highly regulated enzyme, to make sphingosine 1-phosphate, which is, again, itself a very bioactive molecule. And it has receptors in cells, G-protein coupled receptors, 6 of them. And when it binds its receptors, it's really actually 5 of them. And there are other receptors that bind it, but we don't know if they are specifically binding. They're not of they're different family of receptors. But so these are very bioactive, and I'll talk more about them in a bit. But what I'm going to do first is outline where we are interested. We've been really interested in ceramide and sphingosine phosphate, these molecules, because of their bioactive activity, and so we are very interested in the enzymes that make them and break them. And today I'm going to spend most of the time talking about these two enzymes, sphingosine kinase, and a new enzyme pathway we discovered that takes ceramide and sequesters it, so it's out of cell death pathways. And I'll try my best to explain this because I know the audience is very diverse. Okay, next slide. So why these sphingolipids? Again, because of their biologic activity. So I'm going to just show you, they're made by the de novo pathway, as I said, but it's becoming increasingly obvious that cell stress responses induce generation of ceramide in cells, whether it's done by the de novo synthetic pathway or by, before I get to that, whether it's done by the de novo synthetic pathway or by the breakdown of sphingomyelin. So either from de novo synthesis or breakdown of sphingomyelin, you make ceramide. Ceramide, we and others have shown, is a critical lipid to induce cell senescence, differentiation, programmed cell death, and cell cycle arrest. On the flip side, ceramide, as I said, is broken down by ceramidases to make sphingosine, which is then phosphorylated to make sphingosine phosphate by this enzyme sphingosine kinase. And the product, sphingosine phosphate, acts very differently than the first product, ceramide. It induces cell proliferation, mitogenesis, inflammation, migration, angiogenesis, and protects cells from apoptosis. So it's immediately obvious that the balance between those two pathways and these two molecules is really crucial. And so that's why we've been very interested in how this pathway is regulated, and that's what I'm going to spend time about. So I will start by ceramide, discussing ceramide. Ceramide is synthesized in cells, as I told you, by the de novo synthetic pathway. Interestingly, there are 6 enzymes that make ceramide in cells. And they make ceramides, they're called CERS, ceramide synthases, 1 through 6, and they make ceramides of different chain lengths. And I'll illustrate this by this picture. So you can see that the head group is the same in all of them, but the side chains are longer. They're C14 side chain when they're acylated, or C16, C18, which means the length of the fatty acid that acylates. So these long fatty acids are added on to the backbone of the sphingoid to make these longer chain sphingolipids. And there are even longer ones that I'm not talking about today that are found in skin, and also the molecule can be regulated in other ways. So what's interesting about these lipids is they are bioactive, but they also are structural components of membranes. As you see, they're like highly hydrophobic molecules. So why are there so many ceramide synthases in cells? So one of our questions was to try and solve this problem. So a very talented postdoc who is now actually a junior faculty and just got his first R01 with the 6th percentile, so I'm very proud of him, is John Senkal, who actually came up with this idea. He decided that to understand how these different enzymes work, he was going to immunoprecipitate, tag all of them, all the 6, tag them with a flag tag, like a light bulb, and then bring that down and see what other proteins associate, and do they associate differently or the same, so in order to understand their function. So what he showed is, these are the different ceramide synthases, and he showed the red is proteins that associate with each individual one using a proteomic approach. So we studied that using proteomic analysis. And what he found was that one particular protein associates with all of the different ceramide synthases, meaning that it must be important in this pathway. Plus, he found many proteins that associate with different ones, and he's taking these into his work as he moves forward with his own grant. But what's remarkable here is this protein is called fatty acyl-CoA synthetase. So it basically makes a fatty acid. And so this is an enzyme that makes another long-chain fatty acid. They're called very long-chain fatty acid synthetases. So we wondered what the role of this protein is and why is it associating with all these ceramide syntheses. And this just proves that when you bring down the protein, each of your proteins, that's each one of the ceramide syntheses, you immunoprecipitate it, and then you do a Western blot, you can see that they actually all interact at different strengths. So this is a biochemical proof that they are interacting. So what about this protein? So this protein is called long-chain fatty acyl synthetase. Now this, it's a long-chain fatty acyl synthetase, so it makes long-chain fatty acids. And it's an important de novo lipid synthesis, so really critical in making lipids in cells. And it functions in fatty acid uptake, so it's important to take up fatty acids in cells. And it's also found to be highly expressed in cancers, remarkably, a lipid-making protein, highly expressed in cancer. So that got our light bulb got us more excited. And its downregulation in cells, if you knock it down, can induce cancer cell death in lung and glioma, colon, and other cells. Sorry, I forgot, this is out of the frame. But basically, it forms this reaction. A fatty acid plus ATP makes acyl AMP and a phosphate, and then using coenzyme A, it adds and makes an acyl-CoA. So it makes the fatty acid ready to be incorporated into other lipids. So, and what we hypothesized is, since ceramide is made of a sphingosine that's acylated with a fatty acid, we thought this was what it was here for, interacting with ceramide synthase to make ceramide. But we were wrong, and I will not go into great detail why we were wrong, because this is published in a very nice paper by John. He's first author on it, called In Cell Metabolism, and he basically showed through what he did was he modulated this enzyme to see if it changes ceramide levels, and it was doing the opposite. When he knocked it out, it made less ceramide, not more. So we were surprised. So then when we looked more carefully, and I'm making a long story short, but the publication is very accessible for you, and you can understand more. But I just want to tell you how we moved it forward. But what John found that, in fact, this enzyme, ACSL, is interacting with ceramide synthase to add another fatty acid to ceramide. So this is the basic molecule of ceramide, and it's adding another fatty acid to make this huge lipid, which is somewhat similar to triglycerides, because triglycerides that you find in fat are made with 3 fatty acid chains. So this was very intriguing when we made this discovery, and we proved it by knocking out the different enzymes. So you don't have to take my word for it, but we did prove it, and we're pretty confident that this is what's happening. And now we were wondering, so what is the protein? So here you're adding the ACSL, which is the fatty acid CoA synthetase, with the ceramide synthase. But then, but then there needs to be a protein that actually does the transfer, the fatty acid transfer. And so we looked for this protein, and we found that, in fact, to find this enzyme, we looked at what happens with triglyceride, how are triglycerides formed. So from the chemical structure, that to make diglycerides in cells, you have this diacylglycerol, which has it's an alcohol with 2 fatty acids attached and a 3rd hydroxy group. And then if you add this enzyme, diglyceride acyltransferase, which is a critical enzyme in fatty acid in triglyceride synthesis. This is the main enzyme in making triglycerides in cells. So if you add that fatty acids, you make triacylglycerols, which are like all these lipids that we have in our body, and that are possibly regulated by our diet. So we asked the question then, if this looks similar, does actually this enzyme, the same enzyme, diacylglycerol acyltransferase, take ceramide and add an acyl chain, and indeed it did. And this was a very important discovery because no one had implicated this protein except in triglyceride synthesis. So we showed it's actually making acyl ceramide, which is really interesting and different than anyone knew about the function of this protein. And then we wondered, why is it making this, and how is that happening? And so what we looked is that acyl ceramide, when was reduced in 60%, if you have, oops, I'm sorry, this went fast. So if you have the DCAT knockout mice, the enzyme that makes this acyl ceramide, if you knock it out in the mice, actually ceramides the mice are actually eventually are very sick because they can't make ceramides in their skin, and they become very permeable, and they cannot be well because they're losing water through their skin, because ceramides form the permeability barrier. So that told us that this enzyme, when you knock it out, not only does it affect glycerolipid synthesis and triglycerides, but it does affect ceramide synthesis. So we felt we were on the right track. And then in yeast, in Saccharomyces cerevisiae, this enzyme was responsible to make acyl ceramide in that species. So that was another hint for us. So then actually we can say, and again, this is in John's paper, we can say, and we did knockout and manipulation to prove, and then we showed by biochemical studies as well that de novo ceramide synthesized by ceramide synthases that I told you about, the 6 enzymes, all interact with this ACSL5, and they also interact with diacylglycerol acyl transferase to form this acyl ceramide. So this is a new pathway in lipid synthesis that we discovered and we're very excited about. And now I'm going to tell you our newer data that's unpublished about the implications of this. So from that paper, the conclusion was that fatty acid come into ACSL. They interact with ceramide synthesis and with DGAT, diacylglycerol acyl transferase, to make acyl ceramide, and that this acyl ceramide is incorporated into lipid droplets. And this is the proof here that if you transfect cells with the ACSL and the ceramide synthase, you can see here that you're getting lipid droplets in cells, and if you do higher magnification, you see the interaction is at the lipid droplet interface. Similarly, if you transfect with DGAT2 and ceramide synthase, you also see that they interact, and when it's yellow, meaning that the green and red are interacting, that they are again at the lipid droplet interface and in the lipid droplets. So what's happening is when these 3 proteins come together, they make this molecule acyl ceramide, and it goes into lipid droplets in cells. And what's really interesting is that lipid droplets in cells are thought to be people didn't understand what they were. They thought they're just lipid accumulation. But now we know that lipid droplets in cells are very biologically active. They're metabolized, they're regulated, and they're very critical in triglycerides in cells. But we've implicated them here also in harboring ceramide and in taking ceramide to acyl ceramide and incorporating it into lipid droplets. So what are the implications to this? So what we did then, we did a mouse model, and here what we did was we take mice and we feed them either a regular diet or a high-fat diet. And in a regular diet, we take their livers, and their livers are healthy. But if you feed them a high-fat diet, mostly oleate, a fatty acid, they have fatty liver. And then if we inhibit this enzyme, DGAT2, from making the acyl ceramide, in fact, they are protected from fatty liver, and they lose the acyl ceramide, and they make more ceramide. So that was interesting from 2 points of view. First, they regulated cell death, the ceramides that were generated, and also they protected from liver disease. So then we wanted to move this work. Obviously, we work on cancer. We're very interested in cancer. So one question we asked, which people had looked at this enzyme, diacylglycerol acyl synthetase, but no one had ever wondered if it had anything to do with cancer. So first thing we did was we looked at the cancer genome database, and we found to our surprise that many cancers overexpress the enzyme DGAT is highly amplified in these cancers using the cancer genome database. And I outlined ovarian cancer, breast cancer, pancreatic, prostate, and pancreatic. These are all cancers known to have lipid droplets in them. And there are data in the literature to say cancers that have lipid droplets in them are much more aggressive. So we were interested in breast cancer in our group. And so we also looked at the other DGAT, which is DGAT1. And this is even more amplified. And in all of them, the first amplification is from a xenograft cell line of breast cancer. But you can see in pancreatic, ovarian, again, prostate, and breast that they're highly amplified. So we were interested in what's the implication of this amplification. Is that indicating that in cancer these enzymes are working, and what are they doing? And also when we looked at KM plotter to see survival, we find that patients who have high expression of DGAT have a worse survival. So if they express this enzyme and they make a lot of lipid droplets, they have worse survival. So that got us interested in trying to manipulate this enzyme to see if we can rescue cells from cancer. So what we did is, and I'm not showing you a lot of data because there's another subtopic I want to talk about as well, but I just want to give you this highlight information that we took several breast cancer cell lines. These are SKBR3, and they are a HER2 cell line, HER2 positive cell line. We took MDA-MB231, this is a mistake, cell lines, and these are P50 triple negative breast cancer cell line. And we took MCF7 cells, which are epithelial breast cancer cells that are P53, normal for P53. But in every case, what we showed is if you take the inhibitor of DGAT, so the cells are treated with the inhibitor of DGAT either at different concentrations, 0, 25, 50, and 100 micromolar in every case. And you see the white bars that not much happens to the cells when they're treated with the inhibitor alone. But if you add a chemotherapeutic agent, then you are really synergizing cell death, which to us, we know because we measured, what happens is when you treat with this inhibitor, you no longer make acylceramide in cells, so you free up this acylceramide and it goes into ceramide. And ceramide accumulates, and when ceramide accumulates, it acts in synergy with chemotherapy, which we have shown in the past that ceramide and chemotherapy increases ceramide in cells many years ago. But when you add the inhibitor with the chemotherapy, then you are really synergizing and increasing cancer cell death. So for us, this was exciting because it's happening in many breast cancer cell lines and in response to different inducers, cisplatin or doxorubicin. So this for us means that we can enhance cell death in breast cancer cells that are resistant. And what is remarkable is that these inhibitors for DGAT synthesis are already in the clinic because they are being used to inhibit triglycerides in the blood, and they are novel inhibitors of triglyceridemias. They are somewhat toxic because they leave a lot of fat in your gut and they cause GI symptoms, but that can be probably studied better. And the cisplatin and other chemotherapeutic agents are in the clinic. So if you put the two together in a clinical trial, we expect that maybe we will enhance cancer cell death. So that's one of our goals in the near future. And we're working with Dr. Alison Stopik to potentially put a protocol for cancers, like a phase 1. Is that called a phase 1 or a phase something? Anyway. 4 minutes. Okay, so I took too long on this. So I'm going to go into the next part of my talk. Oops, I'm sorry. So the conclusions from this part is ceramide can be converted to acylceramide and stored in lipid droplets, leading to resistance to cancer cell death. And ACSL1 and DGAT interact with ceramide synthases. DGATs are amplified in cancer, and they are important for, if we inhibit them, it's important for cancer cell death. So these are important for implications to cancer treatment. And we're very interested in the mechanisms, and that's the next phase of this work, including hopefully that we would do some more animal and then clinical research. So as I told you, this pathway, we discussed it. One of the other parts of so we've added to this pathway a new addition, which is that ceramide is acylated to acylceramide. So I think the pathway is getting bigger and more interesting. So then I wanted to, in the last few minutes, tell you that mostly sphingolipids are made, like I said, by serine and palmitoyl-CoA, but occasionally, if you don't have serine, or you deprive cells of serine, cells can use alanine, a different amino acid, or they can use different fatty acids. But what happens when, in that case, is, so I'm going to skip all this and just tell you, so this is the enzyme sphingosine kinase that I told you about, and this is just a scheme to show you that it's very biologically active. That these are all the products, these are all the pathways that it's involved in, and it's clearly involved in cell death pathways. So, and it is proteolyzed in response to DNA damage, one way to protect. So what we did is, when you look in cancer, cancer cells synthesize serine at a higher rate, and they require, because they have endogenous some serine, but they also have to make serine because it's not enough for them. And so there was this paper in the literature from the Maddox et al. That showed that when you deprive cells of serine, they are protected and they reprogram cells in cancer. So we were interested because we had seen that our enzyme, sphingosine kinase 1, was degraded in DNA damage in a p53-dependent manner. So when we took p53 out, it was no longer degraded. So because of their paper, we wanted to see what happens in our cells. And we found that indeed, when we take colon cancer cells and treat them deprive them of serine, sphingosine kinase gets degraded. The enzyme that is like very potent in cancer becomes degraded. And we showed that this happens in wild-type colon cancer and in p53 null, but you can see the enzyme is getting lost. So, and that this is mediated by a protease, because when we inhibit proteolysis, we inhibit this breakdown. And I'm sorry that I have to rush through this, but that's when we inhibit this enzyme, sphingosine accumulates, and sphingosine phosphate, the very bioactive molecule, goes down because we're inhibiting the enzyme. I just want to get to the part to tell you that when you form these non-canonical sphingolipids, if you remove serine from cells and use alanine, you form non-canonical sphingolipids that are missing this important hydroxy group that you see in all canonical sphingolipids. And that is really important because this hydroxy group is what gets phosphorylated to make sphingosine phosphate and make bioactive lipids. So when you're missing it, what happens is the sphingoid is no longer able to be phosphorylated by sphingosine kinase, this important enzyme. And apparently, it's been known that when you have things that bind or interact with that enzyme and are inactive, they lead to proteolysis of the enzyme and its breakdown. So we think we know the mechanism why, when we deprive cells of serine, sphingosine kinase is proteolyzed, and it leads to accumulation of sphingosine and to deoxysphingosine, which is this non-canonical sphingolipid. And here I'm showing the accumulation of these non-canonical sphingolipids when you deprive cells of serine. They really accumulate, and they are toxic to the enzyme. So they basically, when they accumulate, when you treat cells exogenously with them, they break the enzyme down. So if you're looking at, and then if you inhibit proteolysis, the enzyme is no longer degraded. So that tells you that they are important in regulating the enzyme. And so we were really interested in this because we wanted to see if now, when we knock down this enzyme by this pathway, what does it do to serine synthesis? And to make a long story short, because of time, and because my timer just went off, I'm going to show you that the final scheme and close. So what we showed in this is that serine, if it's not there, not available, cells can use alanine, and they will make this non-canonical toxic sphingolipid called deoxysphingenin, which leads to proteolysis of sphingosine kinase. So you can no longer make sphingosine phosphate, and the cells become produce reactive oxygen species, inhibit mitochondrial function, and lead to synthesis of serine. So there is a pathway that these molecules, and we're not sure how yet it happens, but they drive PHGDH, which is a key enzyme in serine synthesis. So when you inhibit this pathway, you modulate cells to induce serine synthesis. So I just will close here by saying that we've two important pathways, fatty acids and amino acids, are really critical in regulation of sphingolipids, and by looking at mechanisms, we found that they were important in regulating cell death and cancer. So both have implications to clinical studies, and now I don't show you, but we did some studies in mice where we deprive them of serine. If they have a tumor, the tumors shrink. And so we're trying to study what happens to the sphingoids. So we have modalities that are important in diet that we hopefully can use in fighting cancer. And I will stop by acknowledging my coworkers. My lab group is the following people, and I talked today mostly about John Senkal's work and about J.P. Truman's work. And we have very good collaborators. We have a lipid cancer lab. We all collaborate. And Yusuf Hanoun is always great for advice. So is Dr. Mao and Dr. Luberto. Dr. Snyder helped us with the fatty acid work. Her interest is in fatty acids in the diet. And Jeff Gernon helped us in the serine work. And our core facilities are really crucial. And then my funding sources, and I really appreciate your time. And I'm sorry I went over. Thank you.
Thank you very much, Dr. Obeid. Very nice talk. Now we know that lipids can stress out cells. And when lipids stress out cells, bad things happen. So let's keep that in mind. Our next speaker is going to be Dr. Samir Beyaz. You can come up. Dr. Beyaz got his PhD in immunology from Harvard. Then he worked in Dr. Stuart Orking, who is a known person in epigenetics and regulation of cell death. And he got accomplished through that process and received numerous awards and scientific accomplishments, including the Jeffrey Models Prize. He also got prizes from the American Association of Immunologists, Training Achievement Awards, and a lot of other awards that we don't have to mention. Today, he's going to be discussing the dietary regulation of cancer and immunity. So he's going to look at the relationship between diet and how that influences the immune system that leads to the development of cancer. So thank you for coming.
All right. Thank you. Thanks to the organizers for putting this amazing symposium. There are great talks, and it's a great honor to speak after Lena. I'm a big fan of her. So today I'm going to talk to you about two stories that are seemingly distinct, but they are interconnected. And my lab at Cold Spring Harbor is trying to understand a seemingly simple but really complex question: are we really what we eat? And so this is a question that everyone can comment on, but the problem is there is very little we know about the causality of the associations that we make depending on what type of food we eat and how much they can influence health and disease states. So this is an important problem because what we eat influences not only the way that the nutrients are utilized in our body, but they can correlate with severe pathological states. So obesity is a good model to answer that question, how what we eat may influence who we are. And this problem has been becoming more and more evident both in the US and in the world. So this data is summarizing the incidence of obesity over a decade from 1994 to 2014, and as the color gets darker in red, the incidence becomes higher. And as you can see, it's a self-explanatory slide that it's becoming darker and darker. And this situation is not just limited to the US adult population, it's also influencing childhood, adolescence, and adult obesity rates in the world, in many countries. So, to emphasize why this obesity problem is relevant to this symposium is because there are racial and ethnic inequities that are associated in obesity. So the disparities that are stratified by ethnicities or racial background is summarized in that slide. And as you can see, the incidence of obesity and the rates of obesity, although they are increasing irrespective of the race and ethnicity, there are some differences in rates which are highlighted here in total. In non-Hispanic, Black and Hispanic populations, the incidences are much higher compared to non-Hispanic white and non-Hispanic Asian populations. So there are lots of potential explanations as to why this is happening, and but the problem between these disparities in obesity and the overall increase in obesity rates is a significant health concern. And the reason is obesity is not a single disease, but it is associated with multiple diseases. Examples include cardiovascular disease. Here I'm highlighting the association between obesity and cancer. So many distinct organ and tissue types have significant associations with obesity. What I study is currently colon and rectum, which shows a significant association. And so to understand causal mechanisms as to why obesity or diet-induced obesity may lead to more cancer, we need to divide that question into multiple modules. So the association between obesity and cancer comes from epidemiological studies, and there are significance of these studies, but the causality of these associations are currently lacking. We don't know in particular what cells and what molecules are altering the states that leads to more cancer. And so we don't know to what extent diet is a major player in that equation. We know certain types of diets are pro-obesogenic, such as high-fat diet and Western diet, and societies like the US who consume predominantly these type of diets exhibit higher incidences of obesity, but there are also non-diet mediated influences on that. And so the one key question in my research is to also understand how diet directly can modify the risk of cancer by either altering the cancer intrinsic programs or cancer extrinsic programs. So this is just to again highlight it's a seemingly simple question, but it's a very complicated problem, and there are lots of variables and lack of causality in associations. So we modeled this obesity association to cancer in the context of colorectal cancer several years ago, and now this is a published study where we fed Black 6 mouse long-term lard-based high-fat diet, and as you can see here, the mice got fat after over a year of feeding with high-fat diet, and we analyzed this, the cohorts of mice, and we found that in mice we can recapitulate that epidemiological link that exists between obesity and cancer, where you can see here with red the carcinoma and dysplasia incidences, and with blue is increased. And so this it linearly can be explained like that. So you give high-fat diet as an input, and you get more tumor, but the question mark is a complicated question mark. And so to understand that question mark, I would like to tell you a story. It's called the elephant story, and it's a modern day story now where scientists are trying to identify what this is, and you guys all can see that this is an elephant. So it's the same elephant that we are trying to understand what can cause this high-fat diet mediated or obesity mediated increased risk of cancer.
And so all these scientists are here claiming this is something else based on their point of view and perspective. According to this scientist, it's a wall. So now I will tell you our side of story and how we need to alter sometimes our perspective to discover the elephant. And so to dive into that, I will introduce you briefly what do intestinal cancers actually mean throughout that evolution of the tumor. So intestinal cancers have different stages, and the cell of origin of that cancer is mostly a stem cell that is present in the mucosal epithelium that separates the tissue from the external environment. And so this is, again, you need to make a choice to understand why, when you give high-fat diet to a normal, non-mutated background mice, a high-fat diet, it would give you as an output more cancer. It could be at that stage, it could be at that stage, it could be in all of those stages, but to identify a causal mechanism, you need to be a reductionist and make a decision. So here I will just show you how we study this problem. So the data so far suggesting that most of the time tumor initiation happens due to mutations that are acquired in the intestinal stem cells. And so we took advantage of the advances in this field where, thanks to Hans Clevers and many other people in the field, they identified what are the markers or like the definition of those stem cells inside the intestinal crypt. And here I can highlight to you that the cells that are labeled at the bottom of the crypt, they have that stem cell identity. They are positioned next to their niche cells called Paneth cell. They can elaborate some niche-mediated signals for their maintenance. And so the advancement in profiling these cells and the ability of these cells to be propagated ex vivo in a dish using organoid culture conditions enable us to characterize these stem cells in terms of their activity ex vivo. And so we did that, and I'll just summarize you the key point from that study is the fact that when you take stem cells from a high-fat diet, you see elevation in their stemness. And so this is summarized here. If you take stem cells alone, they are not able to propagate organoids. They need their Paneth cell niche. But when you give the Paneth cells, they are able to form proper organoids. If you take stem cells from a high-fat diet, they have the super stem cell properties. They no longer need niche-mediated signals, which suggest that high-fat diet is modifying that qualitative aspect of the stem cells. So further analysis demonstrated that not only the qualitative aspects of the stemness is increased, but also quantitative aspects of it is increased. Numbers and proliferative potential of the stem cells were much more elevated. And the key data was here the fact that when you give ex vivo lipids to those stem cells in a culture condition, that elevation of stemness was also demonstrated. So the presence of lipids, the lipid-mediated signals in the high-fat diet were probably driving that phenomenon. So to dive into that, we did lots of gene expression studies and identified a key mediator of that process. The mediator was PPAR Delta. It's a lipid-sensing transcription factor. It regulates lots of processes regarding lipid biology, including fatty acid oxidation. I highlighted it here. But what also it does as a transcription factor, it can couple with multiple other transcription factors. And so a key transcription factor we identified that is coupled to PPAR Delta in that condition is beta-catenin. And beta-catenin, as I showed you initially, is associated with stem cell maintenance and activity. And one of the initial oncogenic mutations that lead to colorectal cancer targets beta-catenin program. So APC is one of the earlier mutations that happens in that process. So what we found as a model is in high-fat diet, you get more tumors because your stem cells are qualitatively and quantitatively increased, And the progenitors, together with the stem cells within the crypt, have this elevated stem cell state. So when these are coupled with an oncogenic mutation, they give rise to more tumor compared to the control state. So it's just a statistical analysis that led us to speculate that this is one of the potential contributing causal mechanisms to that process. And how did we determine causality? By demonstrating that PPAR Delta activation was sufficient to mediate that process, and PPAR Delta inhibition was also necessary in that phenotype. So that opened up a paradigm that potentially targeting PPAR Delta in that context, it can be a modality for treatment of the obesity-associated cancers. And for the health disparity standpoint, I think that merits some further discussion. And I would love to hear your points in the panel discussion. So we published this paper, and it was nice to see after our publication that focused on tumor initiation, many other publications demonstrate the significance of PPAR Delta and downstream mediators of PPAR Delta in other tumor models and in other stages of tumorigenicity. So both tumor progression and metastatic potential of the tumor cells were significantly dependent on PPAR Delta and its mediators, and also can be enhanced in response to lipids. So that was a good part of the elephant, I think, we discovered, and it's right here. Now I can say, hey, it's PPAR Delta. But so during my PhD training, I was lucky to get exposed to multiple different disciplines. I was trained as an immunologist. That was a side project that I got drawn into for several reasons. But what I want to tell you is, when you are trying to identify the elephant, which is a very big problem, how diet modifies cancer risk, that's a very, very big problem. And as a young scientist, diving into that question is, of course, ambitious and challenging, but I'm ready to take on that. And when I'm ready to take on it, I want to tell you another type of story where we can identify another part of the elephant by module dissecting the complexities. So the next story I want to tell you highlights the immune aspects in the mucosal tissues. So gut is an immune organ, and there are lots of immune cells, much more than many other parts in your body, and they're constantly interacting with the external environment that are mostly mediated by the gut microbes and the metabolites from the microbes and the food antigens. And so intestine, in that complexity, it cannot be studied just by looking at intestinal stem cells or tumor-initiating cells or tumor cells. So it's a complex network. So the question that I wanted to follow up was how immune surveillance mechanisms are perturbed in response to diet. So we know a lot about immune surveillance mechanisms by now, thanks to the advancements in the field of immunology. We know that in the case of tumor immune surveillance, there is one key adaptive immune mechanism in which tumor cells can present antigens either on MHC class 2 to CD4 T cells or MHC class 1 to CD8 T cells. And the current dogma is the fact that CD4 T cells, through recognition of some peptide antigens on MHC class 2, can help CD8 T cells to release cytotoxic molecules that can kill the tumor cells. But many papers in the field suggested that there are other ways that you can also mediate that tumor rejection, that anti-tumor immunity, and those may involve a direct contribution to the killing by releasing cytotoxic molecules, recruitment of other cells, and through some unknown cooperation between CD8 T cells or like cytokines that may be released to also dampen tumorigenicity. So this is also on its own is an elephant and a complicated problem, but we know that, at least from tumor immune recognition standpoint, tumors try to escape from anti-tumor immunity. And to do that, one of the first things that they do is they downregulate their immune recognition molecules. It's like they get rid of their identity cards and passports. So MHC class 1 is an identity card, and if you don't have it, NK cells can come and kill you. But MHC2 is a passport. You can stay undercover unless you're not traveling around the airports and highly surveyed areas. So we were lucky enough to see that in high-fat diet, a separate mechanism that may contribute to anti-tumor immunity was the fact that a high-fat diet led to dampening of immunomodulatory gene expression in stem cells. So those stem cells, I want to remind you, had an enhanced stemness, enhanced qualitative and quantitative and qualitative aspects in response to high-fat diet. But they also downregulate the genes involved in MHC class II pathway significantly. So we looked into that phenomenon, and since the discovery of MHC class II antigens, both in mice and in human, people have stained the intestinal tissue and have demonstrated that intestinal epithelium expresses MHC class II. So if you look at the textbook, they say MHC2 is only expressed in antigen-presenting cells, but the reality is MHC class II can be expressed in multiple cell types because it's a very responsive molecule to inflammatory cytokines. And so in the gut, we found that in control state, in control diet, most stem cells were positive for MHC class II, and in high-fat diet, this is significantly downregulated. And so the implication is downregulation of MHC class II can be losing that passport, and if those stem cells lose that passport, they can go throughout that tumorigenic evolution faster. So to test this, initially we used the organoid system to understand whether or not cell-intrinsic oncogenic programs are whether or not are altered, and we found that they are not. So we used pre-malignant cells that lost APC and sorted them out based on their MHC expression pattern and found that they are both equally clonogenic in vitro. But in vivo, we developed this orthotopic transplantation system where we can transplant stem cells to the distal part of the colon, and when you transplant MHC positive and MHC negative pre-malignant cells, you can assess their in vivo tumorigenic potential. And we did that, and the result was really surprising and exciting. We found that in the case of absence of MHC2 in a pre-malignant cell, you get much more tumor compared to the control if it is in an immune competent host. But if you do it in an immunodeficient host, that rate was equal. So this is suggesting that the presence or absence of MHC2, which is the passport molecule, is influencing the growth of the tumor depending on the availability of the surveillance system, which is the immune system. So that one is a convincing data, but to show this effect definitively, we developed a transgenic model where we deleted MHC together with loss of heterozygosity model of APC. And in this genetic model, we were able to see that there are much more tumors in the intestine in the absence of MHC compared to the MHC intact counterparts. So there is a relevance in a clinical setting for this phenomenon, and we analyzed TCGA and ICGC data for colon and rectal adenocarcinoma patients, and we found that MHC high expressors survive much better compared to MHC low expressors. So there are lots of contributing factors probably to that survival data, but at least there is a significance that would make us think there may be an impact based on tumor MHC expression throughout the tumorigenic profile. And recently we started diving into this problem deeper, and we have been developing ways where we can, without biasing the system too much, assess how MHC expression can correlate with distinct stages of tumorigenicity. And this is just a brief data suggesting that across tumorigenic program, MHC expression module is dampened. So in tumor or metastatic organoids, we see that there is less and less MHC compared to the tumor. So, but the key question that's remaining is what is the mechanism of MHC expression or downregulation in the context of diet or obesity? And I will show you just two key pieces of evidence, which is based on what we already know about how MHC is regulated. So MHC can be regulated either by pattern recognition pattern recognition receptor activation in response to microbial signals, or cytokine-induced JAK-STAT activation. And we found that, and many other people suggested that in response to high-fat diet-induced obesity, you perturb the microbiome, and this is just showing how microbial diversity is altered in the intestine. And the antibiotics were used as a control, but the intriguing phenomenon we found here, where we stained for MHC2 mRNA using in situ hybridization, is the fact that in high-fat diet and in antibiotic-treated mice, here I can show you in the crypts where the stem cells reside, there is a dampening of MHC2 mRNAs. And so this is the control setting where you see a lot of red, which indicates the MHC expression, and here there is not much. So this suggests that depletion or getting rid of certain microbes is probably contributing to that high-fat diet-mediated MHC downregulation. And so to go deeper into that mechanism, we did microbiome sequencing to deeply identify what are the potential causal microbes in that process. And so on the left side of this plot, you can see the microbes that are depleted, and right side, it shows the microbes that are enriched. And I would like to highlight one of them, the Helicobacter species, are significantly ablated in response to high-fat diet. And the reason that I'm telling this to you is because finding a causal microbe in a microbiome study is really difficult. But I was lucky to come to Cold Spring Harbor because the mice that I used back when I was in Boston were Helicobacter positive. And at Cold Spring Harbor, we have a Helicobacter negative facility. And I have a cohort of LGR5 mice that were in Helicobacter negative, and I compared their MHC expression, because when I was analyzing it, we saw really reduced MHC expression per stem cell. Oh yeah. So in that equation, we are thinking that Helicobacter-mediated modulation of the microbial ecosystem may be contributing to the steady-state expression of MHC2, and high-fat diet is probably altering that ecosystem. I would like to highlight the complication is because we see that if you put a Helicobacter-negative mice on high-fat diet, there is also some downregulation of MHC2. So I think it's good to consider microbiome as an ecosystem rather than a single entity, but at least we have a clue into that ecosystem that Helicobacter may be playing a key role. So to definitively assess these processes, we have been utilizing germ-free mice and doing germ-free add-back experiments, and also doing some mechanistic experiments focusing on the PRR signaling and cytokine signaling. But I want to highlight to you basically in germ-free mice, we see this dampening of MHC2 expression, and when you colonize some candidate microbes that we may think modulating MHC expression, we see the elevation. So to summarize, we are proposing that this tumor initiating stem cell immune recognition through MHC expression plays a key role in anti-tumor immunity, and in high-fat diet, due to dampening of MHC2 in tumor initiating cells, the immune system cannot recognize this properly, and the dampened immune recognition, it's known that it can break the anti-tumor immunity in multiple aspects. We generate those tumors that we are seeing, and I think this is an orthogonal way that high-fat diet can lead to more tumor, and the data is suggesting that the maintenance of MHC2, that passport molecule, is dependent on a healthy microbiome. We don't know what it is yet, but it looks like Helicobacter species are doing something in that equation. And an altered microbiome induced by the diet is doing something that we think it involves a cytokine pathway and also a PRR pathway. And now we are digging deeper into that understanding. But in summary, this suggests that diet-induced alterations in intestinal microbiota can influence the immune recognition abilities of tumor-initiating cells, and that mechanism can be also a potential key causal mechanism behind this obesity-associated cancers. So now we have another part in that elephant story. We thought it was PPAR Delta that does it all, but it looks like MHC also can do it all. So the reason that I told you the elephant story, because we need to be reductionist when we are doing the experiments. You need to choose something and go deeper in that aspect, but also it's good, especially with problems like diet association with cancer or diet association with immunity. These are really convoluted problems, so we need to take a module by module approach while keeping an eye on the other contributing factors. So in my lab, we are trying our best to do that. It's not an easy thing, I can acknowledge to you. So the approach we are taking, dividing these different modules and assessing the phenotypes one by one. So I showed you an example how we can use nutrients such as fatty acids to perturb intestinal stem cells and understand how nutrients can influence tumor initiation processes. We have a huge interest in understanding how nutrients are influencing immune intrinsic mechanisms. I'm giving you an example how nutrients can alter T cell states. And T cell states, I mean T cell migration, T cell activation, T cell memory. And so the black box, which I don't know if I will have enough time to uncover how exactly these things are happening, because we don't have the tools to definitively assess how diet or anything else that can alter the microbiome can causally influence phenotypic states. It's a very hot, popular area, but I think we need to also critically evaluate that area so that we can force innovation to create new tools, new models. So I put the microbiome in the end because I really don't know where to put it, because it can influence everything and can be influenced by everything. So with that, we are trying to use this input-output module, so use nutrients as inputs, and then use perturbation through assessing how these things are altering each module, and then phenotypes that we study, I call them the outputs, and some of them can be relevant therapeutically, as I showed you in the context of targeting PPAR Delta or elevating MHC2 expression, and the end goal is to build a unifying model to uncover the elephant. And with that, I would like to thank people in my steadily growing lab. So Mike left, and he was working with me for 3 years now. He's now a graduate student at Harvard, and Miriam, Highway, Onur, and Brian are my current members, and each of them are diving into a separate question, and hopefully synergistically we will uncover the elephant. I would like to thank Doug. He's my mentor and the leader of the growing immunology community at Cold Spring Harbor. We do some work with Tucson Lab on the organoid models, and Tobias is a recent faculty recruited from UK, and we have been also thinking to expand our research objectives, and I have several other former and current collaborators I would like to acknowledge, and I would love to take any questions, I think, after the, yeah, okay. Well, thank you so much for your attention.
Thank you very much. So we would go to our next speaker, but just wanted to say that was a great talk too as well. So now you can begin to understand how diet actually influences cancer. And this level of detail is really very important for us to understand the underpinnings of these processes so that we can identify the right interventions and studies to mitigate the cancer issues. So our next speaker is going to be Jiyoung Ahn. Jiyoung Ahn is Associate Professor in the Department of Population Health and Environmental Health at NYU Medical Center. Her research focus is usually trying to understand the causes and prevention of cancer in human populations. And she's been very, very active in cancer research, a lot of NIH grants, publications, awards, that kind of stuff. So she's very qualified to give this lecture. So today she's going to connect with what our second speaker just introduced, which is the microbiome. She's going to talk about the human microbiome and diet and how that influences cancer health disparities. Thank you for coming, Dr. Jiyoung.
Hello, thank you for having me today. I want to talk about human microbiota today. Human body hosts trillions of microbiota and create ecosystem. Until last 10 years ago, we were not able to study them because majority of bacteria don't grow outside of human body. So it was really impossible to study them. Recent development of genomic sequencing technology widely opened microbiome area to study comprehensively, including culturables and non-culturable bacteria, and we can accurately quantify them. So totality of microbiota is so-called human microbiome. As a population-based research scientist, my research focus on the established relationship of human microbiome and oral digestive cancer disparity. And unlike small mouse group, 5 mice, 10 mice, humans are more complex. So we think it's important to look at diet and lifestyle factors and how these factors influence on human microbiome and how this microbiome, the diet and cancer relationship is mediated through microbiome. Of course, we do expect some independent effect of diet and lifestyle factors for cancer. My overall goal is to identify human microbiota associated with the cancer. I think this is important because identification of bacteria may lead to personalized prevention by microbiota and lifestyle modification. So this is my talk agenda. First part, I'm going to focus on fairly recent data on diet, gut microbiota, and colon cancer in New York City Asian Americans. Later part of talk will focus on smoking, oral microbiota, and head and neck cancer risk. I become interested in racial disparity in Asian American colon cancer because in general, in native country, colon cancer rate is low. But US Asian American immigrants immigrate to US, their colon cancer incident rate increase, although their rate is still lower than other races. So why is that? I become curious about this. New York City is racially, ethnically diverse. According to New York City Community Health Survey, 43% are foreign-born, including me, and 25% use non-English at home. In particular, Asian Americans are fastest growing racial group in the New York City, account for 13% in New York City compared with average 4% in US overall. This picture shows each dot shows at least 50 people reside in the census area. You do see how Asians reside fairly a lot, particularly in Chinatown and Queens Flushing area. I was really fortunate to be able to develop New York City based on multi-ethnic cohort, so-called family study, food and microbiome longitudinal investigation. We recruited multi-ethnic population healthy volunteers, particularly we targeted Asians, Hispanics, Black, and white. We recruited them through churches, communities, local community center, and local health clinics. Currently, we have enrolled about 5,000 individuals, and we plan to go to achieve 10,000. And this involves prospective follow-up for health outcomes. We ask them to provide stool and oral samples for microbiome assay, and all participants completed demographic and lifestyle questionnaire, as well as diet questionnaire. All materials were translated in multi-languages. So in the beginning, I said the microbiome is assayed by genomic sequencing. Let me just briefly talk about how we did it. 16S RNA gene sequencing is a fairly well-established method for microbiome. And why 16S RNA? This is because all prokaryotes carry this ribosomal component. And it serves as a molecular clock to identify bacterial phylogeny. And this bacterial gene here is fairly small and simple. It contains highly variable regions and also highly constant regions. So we designed the primers to capture these taxonomically identifiable V3, V4 gene regions. And this is a simple overview. From the stool sample, we added an individual barcode and amplified those gene regions, and we sequenced them. What we get is highly paralleling about 500 millions, 500 base pair sequencing read. Based on that, we cluster them based on sequence similarities, over 97%. So each unit is a so-called OTU, operational taxon unit, which is a pseudo-species. So with this OTU, we build a phylogenetic tree based on genomic phylogenic information. And so each individuals we can classify from phylum, class, order, family, genus, and species level. So each one, you will get the information, each individual's bacteria profile per person, per sample. So in the beginning, I said I'm a population-based scientist. This is what we learned over years. So across the whole population, in human population, we tend to share common structure. So here, so everyone in this room, I can tell, everyone will carry Firmicutes, Bacteroidetes, Proteobacteria, Actinobacteria, Fusobacterium. These are all common. We share these common members. However, each individual, we do have each individual variability per each content. So it's probably you cannot read, but this is like 1,000 individuals bacteria profile in genus level. You do see somewhat variations. So my bacteria content, overall microbiome content, is not necessary with Dr. Lee's microbiome profile. So we do have some individuality. So this is inter-individual variability confer disease susceptibility. And what are the determinants of this inter-individual variability? So before I talk about family study, I want to go back to several years. In 2013, we conducted a case control study of colon cancer and gut microbiome. We recruited 100 colon cancer patients at the clinic, and we also recruited age, sex, race matched health non-cancer control group. We simply compared their microbiome. And this is what we learned. Cancer groups, so this clodogram is a nice way to summary, and the cancer groups shown the red. Cancer groups are 2.5 times more likely to carry Fusobacterium compared to control group. And the second point is Clostridia, which is commensal bacteria, was significantly depleted in cancer patients compared with the control group. Other groups, basic scientists, reported that Clostridia play an important role fermenting diet fiber to butyrate, and which is known as an anti-carcinogenic component. Later, one of my fellow, Dr. Lin, showed that high fiber consumers tend to have high Clostridia abundance in healthy population. So let's go back to the family study. This is what we learned. So about 1,000, we sequenced about 1,000 those people who participated initially, early participant in family study. We sequenced the microbiome. In particular, Dr. Peters, my postdoc fellow, she received a pilot funding from postdoc funding to looking at the Asians and white gut microbiome differences and acculturation. So this PCOA plot is a nice summary of showing the microbiome differences. So each dot is called a Johnson-Shannon Diversity Index, which is a summary index. I'm not going to tell you all the formula, but in a just short word, each dot summarize each phylogenetic profile, and each two dots are close. That means their microbiome phylogenetic distance is close. They do have a similar microbiome profile. If dots are apart, that means their microbiome profiles are very different, overall structure. So what we've seen is a striking difference between Asians, shown by blue dots, and then non-Hispanic whites, shown in red dots. And now, I apologize for this small font. So now, you want to know what specific bacteria contribute for these gum microbiome differences. What we learned is particularly Clostridia abundance was particularly different between Asians and whites. So we found the colon cancer related bacteria. And obviously, it's known for fiber metabolizing bacteria when we compared the fruit and vegetable intake in Asians and whites, and Asians tended to eat more fruits and vegetables. Well, so this is predetermined with this. But what about, is it really like a race, or would it be more, what about without just knowing the determinant? Why don't you just to analyze this data differently? She did unsupervised clustering analysis, and then we found there are 4 separate distinct microbiome groups. So here, so here the first blue reached in Korean population, shown in the blue here. And this one, the second interesting group is pink group, and primarily non-Hispanic whites. And green and purple, they are at the mixed group. So again, it's another way of visualization. And we measured comprehensive diet intake with food frequency questionnaire. And we do see that Korean-rich clusters are associated with high seafood, tofu, soups, cooked grain, fruits and vegetables, versus this white-rich group is strongly related to high cured meat, poultry, fats, and oils and cheese. So this is still ongoing analysis, fairly new, working on process project, but we realized this race is not probably black and white. There is an acculturation process going on. So we are currently looking at what are the bigger determinants of this diet acculturation. So we are looking at social, cultural, neighborhood impact in this New York City resident diet acculturation and microbiome effect and possibly cancer effect. So our tentative summary is East Asian immigrants increase colon cancer risk after moving to US. Lower gut Clostridia may be associated with higher risk for colon cancer. Asians have different microbiome, at least partially explained by diet and bacteria, and we are still working on understanding diet acculturation among Asians. Now I'm going to moving on to my second talk, smoking, oral microbiota, and head and neck cancer risk. A while ago, several years ago, I showed that people with moderate and severe periodontal disease, which is inflammatory gum oral disease, is 2 times more likely to develop and die from upper GI cancers including head and neck and esophagus cancer. This is based on secondary data analysis of CDC and Haynes data. Why is it? So my hypothesis is this could be due to something oral bacteria. Subsequently, we obtained the NCI funding. We were able to study oral microbiome and these upper GI cancers. The way we approached was we used the prospective study. This is in collaboration with the NCI and American Cancer Society. When I was in fellow at NCI 15 years ago, NCI developed this population cohort. And this NCI and ACS recruited 140,000 healthy individuals. And they collected, when they were healthy, they collected mouthwash samples. That time, mouthwash samples were designed to get human DNA, but now we are using for the microbiome study. So those 140,000 healthy individuals were followed up over 10 to 15 years. During this follow-up, some people developed the cancer, majority of people never have developed the cancer. So we selected those prospectively developed 130 head and neck cancer cases and 106 esophagus cancer cases. And we also selected age, sex, race matched, cohort nested controls, those who never developed cancers. And this is important because we can study bacteria profile when they are healthy, and we can see which bacteria profile is associated with the subsequent development of these cancers. So we completed the essay. This is what we have found for head and neck cancer. From this, this is a beta proteobacteria group. We learned that people who carry more proteobacteria in their mouth associate with 30% less likely to develop head and neck cancer subsequently. And this is first study to show the direct link of oral bacteria and head and neck cancer. At the same time, we also showed that this same bacteria is inversely related to esophagus cancer development, although effect size is a little smaller than head and neck cancer. So now you might wonder what this bacteria does. And we didn't test the function directly, but we created the inferred metagenome profile. In that way, we can see which genes each bacteria carries, and we can group them by CAG gene functional pathway. So we learned that these beta proteobacteria groups play an important role in xenobiotic biodegradation pathways, including chlorhexidine, nitrotoluene, styrene, toluene degradation. You might wonder, so what? And these are important smoking carcinogens. Now, so then we decided, well, that if that is true, then this relationship would be more pronounced in the high carcinogen environment. So we divided our group, former, never, current smokers, and we evaluated the relationship of proteobacteria and head and neck cancer. As expected, we found the inverse relationship of proteobacteria and head and neck cancer were more pronounced in current and former smokers. So this is a summary. Greater oral beta proteobacteria abundance decreased risk of head and neck and esophagus cancers. Beta proteobacteria may involve in metabolizing carcinogens. And this may lead for targeted prevention for beta proteobacteria, particularly in smokers, while encouraging them to stop smoking. So today I just talked about, my talk was focused on cancer development area, but there are also important areas we are working on is cancer progression. Those people who developed and will be treated, whether these bacteria play a role involves for the treatment response and progression. For example, melanoma immunotherapy, those people who treated it, whether gut microbiome predictors for response to therapy. We are actively collaborating with other area of scientists. For example, we identify bacteria. These bacteria need to be tested in basic science for the mechanism. And another group of collaborators is actively working on identification of developing probiotics and specific antibiotics. And lastly, we are actively collaborating with method development experts like statistics and laboratory. So we are in the very exciting area times, and we just begin to understand about human bacteria and cancer relationship. So why this is important? And identification of bacteria may help to learn about the causes of cancers and may help us to identify people at high risk for cancers. And this may lead to ways to prevent cancer by tailored microbial and diet lifestyle modifications. So this is truly based on teamwork, and we have great collaborators at NYU, CDC, ACS, and NCI, and I am grateful for funding support from NCI for make this project happen. Thank you.
I need the speakers to come up for question and answer. I'm Jenny Williams from Stony Brook University, and I'm going to moderate this section. The problem that I had today was the weather. I came here, I left my house at 6 a.m. Siri told me that I would be here at 7:30. It did not happen. Okay, I would like to actually ask the first question, Dr. An. I actually was interested in the clustering that you showed for the Asian population. And I know that with all studies that we have done, there are inter and intra differences between races and ethnicities. My question to you is that you had these clusters. Were you able to see any differences in the microbiota between one region and another region? Did you actually do that test? Or?
You mean geographic region?
Excuse me?
You mean the region with a geographic region?
Yes.
Yeah, so this is, it's really, we got the data last week, so we really didn't have a chance to explore deeply, but it's those Korean population fairly, we recruited from the Queens, those area. And we have a few Asian Americans reside in Manhattan, but we didn't really look at yet. But I think it's a great question we can look at, yeah.
Okay. Is there anyone here have a question?
Anybody?
We have a question back there.
Is there someone closer there? Oh, sorry.
Samir, I have a question for you. So, which is a very great presentation. So, one question for you is: have you taken a look of the PPAR alpha and the PPAR gamma, because those are also very important for the metabolism, especially in the high-fat diet condition?
Yeah. So. So the predominant member of PPAR family that's expressed in the intestine is PPAR Delta, and it's highly enriched in the stem cell compartment. But so in vivo, when you delete PPAR Delta, you see some compensatory mechanisms that still maintain some of the effects of high-fat diet. So we did delete PPAR Delta in a background of PPAR alpha knockout mouse, and so I think there is some sort of interaction in those two modules. PPAR gamma doesn't seem to be expressed that much in intestinal stem cells. It's shown to be expressed in some other cell types throughout the intestine, and I think inflammation may promote its expression state or activity, but we haven't looked at it. But I know some other people have done some work on PPAR gamma and intestinal epithelium.
We have
Hi.
one question over there.
Thank you for the great talk. I have a quick question for the panel, especially Dr. Reyes. In terms of the ketogenic diet, there has been preclinical studies that have shown anti-tumor activity, and we know with ketogenic diet, it's a low-carb, high-fat diet. Do you have any comments regarding that in terms of effects on the tumors, and are you looking into that in future studies?
Is it for me or for?
I think for everyone.
Oh, for everyone.
Do you have comments on ketogenic diet?
Actually, it's a very good question. So in ketogenic diet, I guess you have to know what exactly is in that diet, right? Because sometimes it's a high protein, sometimes it's a high fat, sometimes it's a combination. So if it's a high fat ketogenic diet, then it would be really interesting for Samir's work. And it would be also interesting for us because we, as I said, in collaboration with Dr. Snyder, where we fed mice high-fat diet, and we found that when they had they got fatty liver that was inhibited by that inhibitor of the acylglycerol acyltransferase. So, and we also saw in cancer that if it is a high-fat diet, the ketogenic diet, then it might have important implications to cancer for us, but would be something worth trying for sure.
So like in the story that I showed, we used lard-based high-fat diet. But so if you look at the key genes that are upregulated, they are also like some of those genes activate ketosis and ketogenesis, and also imply that there is some ketone bodies in the system. So the effect of ketogenic diet on cancer as a direct input is a very complicated problem, because then it will also deal with, as Lina alluded to, is the type of and the types of the fatty acids that may be enriched in the way of you are utilizing that diet. Mechanistically, there are, like Luke Hentley and others have been promoting that idea in the context of PI3 kinase mutations, which may have some mechanistic connection, but I don't know yet whether or not within the phenomena that I described to what extent ketogenic diet can be protective and high-fat diet can be pro-tumorigenic. That's why I use the elephant analogy. We are just pointing out individual isolated incidences in a very nice controlled experiment that you are doing in the lab. So it's very difficult to go and make big claims saying that high-fat diet would cause cancer and ketogenic diet would save patients from cancer. So I cannot make any claims, but we are doing for the microbiome part to understand whether or not is it the types of the fatty acids, or is it the type of the diet or the state of obesity that's influencing MHC expression. So far it looks like it is, if you have high fat, regardless of the type of diet you do it, you see dampening of MHC2. Yeah.
Do you have a comment or no? So the other thing I realized right away also, if it's a high protein diet, then as well as high fat, then of course, as I mentioned, when you have like cancer cells are addicted to serine, they're all amino acid. They're also addicted to glucose. And I suppose they are to a certain extent like fat a lot. So I think if you have 2 or 3 of the above, I think it's not great. And I think as a geriatrician, the only thing that's been shown to prolong lifespan is low-calorie diets, which are low-fat, low-protein, low-calorie total, and they prolong lifespan in all species, from worms all the way to humans. So the alternative is not fun, but that's probably the only thing that you can do.
One more question back here.
Hi, Steve Itzkowitz from Mount Sinai. For Samir, I'm intrigued by the difference between the small intestine and the colon. The small intestine never gets cancer, and the colon often does. And your model with the intestinal stem cells and the Paneth cells, that's applicable to the small bowel. And you showed that you can actually replace the Paneth cell function with high-fat diet. A question is, the organoids that you work with, and forgive my ignorance about this, are they derived from colonic stem cells or small intestinal stem cells? That's one question. The other question is, obviously, the high-fat diet gets broken down and metabolized on its way down to the colon. What kind of high-fat diet metabolites are present in the lumen of the colon that are different than the small intestine?
So those are two great questions. So the first one is the stem cell model was reductionist, right? So we had to focus on small intestine because the APC LGR5 model that we chose is giving measurable effect sizes in the small intestine. So the biology of the LGR5 mediated stemness and the niche is elusive in the colon. There are some recent understandings on how the stemness and the niche is present in mouse colon. So it's more defined in the intestine, and that's why we chose to focus on the stem cell assays using stem cells isolated from the intestine. But in that paper, of course, we also did the colon and showed that colonic LGR5 cells from a high-fat diet fed animals also demonstrate increased stemness. But I think the rules of lineage and the orders of differentiation are a little bit different. So that's something that I should acknowledge. For human and mouse, there are also stark differences because in mouse, when you delete APC and the most current models like APC min or LGR5 APC would give you small intestinal tumors predominantly. But in humans, you see rarely these effects. And there are some theories out there, and there are big discussions whether the identity of the stem cells or division or diet. Yusuf's group published a really nice paper to provide another perspective on that equation. What I can just say, our finding that the stemness as an entity is enhanced in response to a high-fat diet has some molecular rationale through PPAR beta-catenin interaction. And so if you elevate beta-catenin just by focusing on that pathway of stemness, I can predict that that is going to happen also in the colon, but we are limited by the model that we chose. So we were discussing with some colleagues, if you use DSS and AOM mediated colitis, whether we are going to see the same thing. We haven't done that experiment, so I don't have that clear answer. And so the second question, I think, is also a very interesting point of view. But we still are actually working on trying to understand that complexity. So all these things are part of that elephant still, and I think we'll spend some years ahead of us to answer some of these questions.
Okay, we have one more question over here.
Just a question for Dr. Ann. Did you stratify when you were comparing the microbiota from the immigrants, and did you stratify according to how long they were here? Because, like, I mean, one would assume if you can be an immigrant for the 2 years, you could have been an immigrant for 30 years, and you would assume that your diet changes the longer you are here, and with that, your microbiota and everything would change.
Right. So when it's actually, the answer is we didn't have the information. It's because the reason was when we developed this cohort, we learned that a lot of minority population, it's a lot of undocumented population, is actually our participant, a lot. And so those are very sensitive information. So we had a lot of debate and whether we're going to ask this, but we decided not. But eventually, probably in the follow-up questionnaire, if they want, probably we can ask, but currently we don't have that information. But I think that's key. We thought, I really realized after that that's really key information to, it's missing in this analysis. But on the other hand, it's not probably, we learned from social scientist collaborators. It's not just, yes, year of US reside is important, but it's not like a actual time. Each individual has a different level of acculturation. So maybe that information can be replaced by actual acculturation index. Does that answer your question? Yeah.
Okay. So, excuse me. Okay, we are going to take a 10 minutes break, but before we go, give a hand to our speakers.